Hi guys, what's up? Today we're going to talk about biotransformation. Biotransformation. So we're on the pharmacology series, right? So we're on the next topic, that's biotransformation. The first step in pharmacokinetics was absorption. After that, the drug got distributed, and now the drug is going to be metabolized, or in another word, we can call this biotransformation, or simply metabolism, right? So, what is metabolism or biotransformation? So, when you take a drug, for example, this is a drug, and the drug gets into your body. Now the drug gets active like enzymes, right? And the enzymes convert the drug into metabolite. Now this whole process is what you call biotransformation or metabolism. So when we talk about metabolism or biotransformation, we tend to think that metabolism is a process of converting a drug from its active form into inactive form, right? So when we talk about metabolism, we tend to think that a drug is getting converted from its active form into the inactive form, but that's not true always. Yeah, that's right, that's true in 80 to 90% of the cases, but that's not always true, because there are many other cases where the drug gets converted into the active drug gets converted into active metabolite, right? And there are some instances where the drug can get converted into from prodrug state into the active drug, or there are some cases where the drug gets converted into toxic metabolite. So now you see, drug gets converted into inactive form. This is true in 80 to 90% of the metabolic process, but in metabolic process we can also see that the drug is getting converted into from active form into active metabolite. And again, the drug is getting converted from the prodrug form into the active drug. Again, the drug can get converted from the drug into the toxic metabolite form. So is it correct if we tell the metabolism is the process in which the active drug is getting converted into the inactive form? Is that right? Not actually. That's not right. So the more correct definition of the metabolism to include all these things is the metabolism is the process in which we convert a drug from its active form into its more water-soluble form, right? So if we tell this definition, then this would be more better or more correct. So now, I think by now you have understood the concept of metabolism. Now let's get into the different types of biotransformation that we have to know from exam point of view, right? So in metabolism, there are two different kinds of reactions, right? So there are two different kinds of reaction. That's phase one, and phase two. You know what? The term phase one and phase two is not actually correct, because there's actually type 1 and type 2 reactions. Because whenever we tell that phase 1 and phase 2 reaction, we tend to think that the phase 1 and phase 2 reactions are the reactions that occur in sequence, but that's not right, because it's actually type 1 and type 2 reactions. And you know what? This phase 1 and phase 2 reaction that can occur in any sequence. The phase 1 reaction can occur the first time, or the second reaction can occur the first time, or both reactions can occur at the same time. So rather than calling phase 1 reaction or phase 2 reaction, it's better to call type 1 and type 2 reaction. But by convention, we're using the word phase 1 and phase 2 for now. Okay, so let's talk about the phase 1 or type 1 reaction. So phase 1 or type 1 reaction. So what's phase 1 and type 1 reaction? Whenever we talk about phase 1 and type 1 reactions, first three words that has to come in our mind is oxidation, or reduction, or hydrolysis, right? So phase 1 reaction or the type 1 reactions are those reactions which involve the oxidation, reduction, or hydrolysis. So under this reaction, what are the reactions that involve reduction and oxidation? Basically, the reaction which involves oxidation that also involves reduction. So oxidation and reduction, this redox reaction cannot be isolated. So whenever we talk about oxidation, reduction reaction also comes together. So oxidation and reduction reaction, the example of these reactions can be the reaction which uses cytochrome P450 enzymes. Reactions involving cytochrome P450 enzymes. Right? Now, another type 1 or phase 1 reactions that comes under the oxidation and reduction reactions next to this reaction involving this cytochrome P450 enzymes are, reactions involving monoamine oxidase enzymes. Right? Now, there's another example of the oxidation reduction reaction, that's alcohol metabolism. Right? Now, these are the things that come under oxidation reduction reactions which fall under type 1 or phase 1 reaction. Now, what is next? Hydrolysis reaction. There's also hydrolysis reaction that involves, we'll talk in detail when we come to the topic. For now, just remember that there is hydrolysis reaction which falls under the type 1 or phase 1 reaction. Right? So out of these reactions, from example point of view, this reactions involving the cytochrome P450 enzymes is very, very important. So let's talk about this. So let me erase this first. So when talking about this cytochrome P450 enzymes, there's some basic things we have to keep in our mind, and that I'm going to draw in a table. So we're talking about the cytochrome P450 enzymes. So there's some basic information you have to know. First of all, let me write your cytochrome P450 enzymes. Right? Now, there's a term called substrate. Now, there's next term that's called inducers. And now there is next term we call inhibitors. So what is cytochrome P450 enzymes? There are actually, there are hundreds of enzymes, maybe about 100 different kind of cytochrome P450 enzymes you can find. So you can always memorize all these things. And these 100 different enzymes are, put in 14 different families, right? Now these enzymes, each and every enzyme has their different substrates. For example, this is your substrate. And when the substrate is acted up by the enzymes, the substrate gets converted into the metabolite. I think everything is clear to here now. So you're now clear with the concept of substrate and what substrate means. And the substrate is acted on by the enzymes, and the enzyme and the substrate, when they react together, that converts into metabolite. Now, what is inducer? So before getting into the concept of inducer, first you have to know what normally happens in a cell. For example, this is your cell, right? So, and this is the chromosome that is present in the nucleus of the cell. Now, inside this chromosome, suppose this is your gene. Let me use another color here to make it more interesting. So this is your gene, right? Now, suppose this gene is getting expressed. When this gene gets expressed, suppose this gene's expressed and produces an enzyme or a protein, and that enzyme happens to be B450 enzyme. This is how cytochrome B450 enzyme is normally produced by the cell. This is a cell, and this is the chromosome inside the nucleus of the cell. Now, this is the gene in the chromosome. Now, when the gene is expressed, the enzyme is formed. This is how the cytochrome P450 enzymes are formed. This is what normally happens in the cell. Now, when you give inducer in the body of the patient, suppose you give inducer to the patient. Now, what the inducer does is, as the name suggests, the inducer induces or it increases the gene expression. When the gene expression is increased, what happens is more amount of the enzymes are formed, right? So, in simple word, what you need to understand is the inducer, when it gets into the body of the patient or a person, or when it acts on the cell, the gene expression is increased. When the gene expression is increased, the enzymes production is increased. Now you see, when the enzyme is increased, the substrate are metabolized in higher rate, right? So the concentration of the substrate now goes down in the blood. For example, so before getting into that, let's talk about inhibitor. So we talked about cytochrome P450 enzyme in just basic terms, right? And we know about substrate, right? What is substrate? Now we know what is inducer right now. What is inhibitor? Inhibitor. So I already told you that this is the normal thing that happens in the cell. Now, in place of inducer, what if this is inhibitor? What if this is inhibitor? So if this is an inhibitor, what the inhibitor does is just opposite to the inducer. The inhibitor inhibits the gene expression. When the gene expression is inhibited, now the production of the enzyme is decreased. Now when the enzyme is decreased, let me use another color to make it more interesting here. Let's see here. The enzyme is decreased. Now the enzyme's amount is decreased in the blood. Now the substrate cannot be metabolized that much. So the substrate is not being metabolized that much. Now the substrate's amount increases in the blood. So now you see the contrast. When the inducer is high in the blood, the substrates are metabolized in higher rates, so substrate's level in the plasma goes down. And when the inhibitor is used in the plasma or the blood, what happens is the substrates are less metabolized. Now the substrate's level goes up in the blood. So in gist, what I'm talking about is the inducer. The inducer, right here, the inducer causes increase in plasma concentration of a drug. Decrease. The inducer increases the enzymes amount, and more enzymes and more substrate are metabolized. So a less amount of substrate in the blood. And if there's inhibitor in the blood, what it does is the inhibitor inhibits the enzyme, right? When the enzyme gets inhibited, less enzyme is available. Less enzyme means less substrates are getting metabolized and more amount of substrate remain in the blood. More amount of plasma, more plasma concentration of a drug in the blood, right? So this is the thing we need to remember from exam point of view. Only this. Now, we know about cytochrome P450 enzyme, we know about substrate, we know about inducer, and we know about inhibitor. Now, let's put one example here. Very, very important example from exam point of view. Suppose, in place of theophylline, in place of cytochrome P450 enzyme, let's take an example of one enzyme 1A2. Now, in place of substrate, let's take an example of a drug that is very dangerous when we use in high dose. That's theophylline. Theophylline, right? Now let's take an example of a drug that's macrolide. Sorry, no. Let's take an example of an inducer, for example, cigarette smoke. Cigarette smoke basically contains the hydrocarbons, and these hydrocarbons act as the inducer. Now let's talk about one inhibitor that is macrolide. So let's take an example of macrolide, that's azithromycin, for instance. Now, suppose a person is a chronic smoker. When he smokes, there's hydrocarbon in the smoke, right? So when the inducer or the smoke gets in the inducer, the smoke, in actually the hydrocarbons and it's present in the smoke gets into the blood, what it does is it induces the gene expression. So the more amount of more gene is expressed and more amount of enzymes are produced. Now, more enzymes and more substrate are metabolized, right? More amount of substrates are metabolized. Now you see the theophylline. Now you see the theophylline, more amount of the theophylline is metabolized here. So the theophylline concentration in the plasma goes down. So from all these pictures, what we need to conclude is that in the smoker who is very smoking chronically, so the level of the enzyme that metabolizes the theophylline goes up, so the level of theophylline in the blood goes down. So in case of chronic smoker, if we have to give theophylline, we have to increase the dose of the theophylline in the patient. Now, if we take an example of inhibitor, if we use the inhibitor macrolide, or for example azithromycin, the azithromycin will decrease the gene expression. When the gene expression is decreased, the enzyme production is decreased. Now what happens is less enzyme, the substrate, less amount of the substrate is metabolized. Now you see more amount of theophylline remains in the blood. Now you see more amount of theophylline remains in the blood, and more amount of theophylline in the plasma of the blood means it can show its side effects. So that type of patient may come into the emergency with presentation of arrhythmias, which can be very dangerous to his life. So if a person is a smoker, he may need high dose of theophylline, right? And if a person is taking azithromycin-like drugs, which are inhibitor of the cytochrome P450 enzymes involved in the metabolism of the theophylline, then we have to consider giving him very low dose, because the high dose of such type of dangerous drug may kill him, right? So just as a bonus point, what is the condition when you want to give a patient azithromycin and the theophylline together? In situations like chronic bronchitis, right? In situations like chronic bronchitis, what happens is in chronic bronchitis there's excessive production of the sputums, right? Mucus. So when there is excessive amount of the sputum in the respiratory tract, there is a general rule in pathology that whenever there is a blockage in a tube, the places where the locations behind the blockage becomes a conducive environment for organisms to colonize that place. So if here is blockage due to the overproduction of the mucus, then the places distal to this can be a shelter for organisms. Now the organisms can cause infection by colonizing that place. So to prevent the infection, we give azithromycin. And the patient with chronic bronchitis or the patient of COPD, we give theophylline because that's a bronchodilator, right? So the patient is under theophylline, and for the patient to prevent infection, we're giving macrolide. without thinking the danger or interaction of these two drugs. Now, what happens is the azithromycin we just give is decreasing the enzyme amount, and less enzymes, less theophylline is being metabolized, more theophylline is getting accumulated in the blood, and more theophylline, and the theophylline shows its side effects. And the dangerous one is arrhythmia. So you may kill the patient unknowingly, right? So this is the thing we need to consider when you talk about the cytochrome P450 enzymes, and these are very high yield points from exam point of view. So now let's talk about some very famous examples of the inducers and inhibitors. So to remember this, inducers and inhibitors, there's a very interesting mnemonics that I'm going to share you, and that's kind of funny. So for inducers, there is mnemonic that goes like, I found this mnemonics in some books, so it's not my own creation. So smoking and drinking in bars, car, riffs, or fan. I don't know what this sentence actually says, but for a mnemonic, I think it makes some sense to remember these things, right? So this smoking means it's referring to the hydrocarbons that's present in the smoke, and all drinking, that means it's referring to ethanol, right? And bars means barbiturates. It's referring to the barbiturates. Now, car is referring to carbamazepine, right? And riff means rifampin, and fan means phenytoin, right? It's just a way to remember the drugs. Now, what is the next way to remember the inhibitors? So the next funny mnemonics is COK Coke plus grapefruit juice. Grapefruit juice with pie. You may add apple pie because that's if that makes you the mnemonics more interesting, right? So this C here refers to cimetidine, and this O refers to omeprazole. This K refers to ketoconazole, and this E refers to erythromycin. And this grapefruit juice is also a very famous inhibitor because the grapefruit juice contains a substance like furanocoumarins. So the furanocoumarins can act as an inhibitor because there's one interesting vignette they can give in the exam. For example, suppose there was a patient that was in a follow-up, and he was a patient of hypercholesterolemia, and he was atorvastatin. Now, you counsel the patient, you should change your lifestyle if you want to live more. And while trying to change the lifestyle, he now started to using herbs, and he started using or drinking the fruit juice. Now, while choosing the fruit juice, he personally liked grapefruit juice. Now he's taking atorvastatin at the same time, and he's also now started drinking the grapefruit juice, which contains the furanocoumarins. Now, what the furanocoumarins can do is, this is an inhibitor. Now, the inhibitor inhibits the enzyme production, right? It inhibits the gene expression, and the enzyme production is decreased. Now, what happens is the furanocoumarins present in the grapefruit juice has decreased the enzymes, right? And now, less enzyme, less amount of the atorvastatin is metabolized in the blood, right? So more amount or high concentration of the atorvastatin is present in the blood now. So the patient may come with a complaint of muscle pain or muscle soreness that can be the side effects of the atorvastatin. So you never ever prescribe a patient arovastatin and grapefruit juice together, because grapefruit juice drinking can be a means or way of changing lifestyle of a patient who don't know their interaction. So always be aware. We have to be aware of this. Now, and PI. What's PI? PI means protease inhibitor. Under protease inhibitor comes the drugs that we give in the patient of SIB, like the ritonavir or indinavir, like that. So these are the things we need to talk about in cytochrome P450 enzyme section. Now let's get into another type of hydrolysis and oxidation and reduction reaction. So till now we have covered. We'll talk about biotransformation, right? Under biotransformation, we talked about the definition of biotransformation or metabolism right now. Then after we talked about type 1 or type 2 reaction, or phase 1 and phase 2 reaction. Now, under the phase 1 reaction or type 1 reaction, we talked about the different kinds of type 1 or phase 1 reaction that can be oxidation or reduction reaction or hydrolysis. And under that oxidation or hydrolysis, we talked about this reaction that can involve the enzymes, cytochrome P450 enzymes, right? Now the next reaction that come under the oxidation and reduction can be the reaction that can use monoamine oxidase. So one very high point in the reaction that involves monoamine oxidase is, let me write in the subtopics here of type 1 reactions that can be oxidation, reduction, or that can be hydrolysis. Hydrolysis, right? So under oxidation and reduction, we talked about cytochrome P450 enzymes involving reaction. Now we're talking about monoamine oxidase enzyme involving reactions. And in this type of reaction, there's one very important point we need to discuss. So suppose this is our substrate. This is MAO, and it converts the substrate into metabolite. The only important thing we need to discuss in this lecture is what are the substrate that MAO is going to metabolize. They can be endogenous. They can be endogenous neurotransmitters. Neurotransmitters like serotonin, or that can be dopamine, or that can be norepinephrine, right? So the next substrate of monoamine oxidase can be exogenous compounds. Exogenous compounds like tyramine, right? So for extra knowledge, if I ask you what are the things in which we can find tyramine, these are the wines, these are the fish, these are cheese. We're gonna find a substance called tyramine. This is an exogenous compound that is also metabolized by monoamine oxidase enzyme. Now, this is the only point we need to talk about monoamine oxidase enzyme involving reactions. Now, let's talk about next oxidation and reduction reaction that comes in the phase 1 or type 1 reaction. And that's alcohol metabolism. So alcohol metabolism also happens to the phase 1 or type 1 reaction. So what happens to alcohol metabolism? The alcohol gets converted into the aldehyde, and the aldehyde gets converted into the acid, right? So these all reactions are proceeded by, or these all reactions are helped by dehydrogenase enzyme, or catalyzed by dehydrogenase enzyme. So basically what you need to know is alcohol metabolism is also oxidation or reduction reaction, which comes under the phase 1 or type 1 reaction. Now, there's another type of reaction, that's hydrolysis. So under hydrolysis, what are the reactions that we need to know? First, let's talk about what is hydrolysis. Hydrolysis is basically breaking the bonds present in the drug by using water molecule in presence of enzyme. For example, this is drug. Suppose in drug there are different elements, right? So suppose these are the bonds present in the drug molecule between different atoms. Now if we add water here, and if this reaction is catalyzed by enzymes, right? So in presence of enzyme, when you add water in a drug, then the bonds between the drug molecules get broken. So this reaction is called hydrolysis. So hydrolysis is basically breaking the bonds between the atoms in the drug, or breaking the drug molecules, bigger drug molecule, into simpler form by using or by adding water molecule in presence of enzyme. That's basically hydrolysis. I think you got it now. So what are the example of hydrolysis reaction? For example, succinylcholine. So hydrolysis, the best example can be the metabolism of succinylcholine. It gets converted into metabolite, and here is some enzyme involved in hydrolysis. Some enzyme called esterase. This esterase enzyme helps the succinylcholine to be broken down into a simpler form. And one very important thing to know about these enzymes has polymorphism. So when some enzyme has polymorphism, the enzyme that I have in my body can have different metabolizing capacity than the enzyme that you have in your body. Same enzyme but can have different capacity to metabolize the drug because genetic polymorphism can occur, and same thing can happen in the dehydrogenases that are involved in alcohol metabolism, right? So genetic polymorphism, the capacity capacity to metabolize the succinylcholine can be different in different patients. Now let's talk about next reaction. And that's type 2 reaction. So we talked about phase 1 of type 1 reaction. Right now, it's turn to talk about type 2 or phase 2 reaction. So what is type 2 or phase 2 reaction? This is there's another name we can use for this type 2 reaction, and that's conjugation reaction. Right? So why we call this conjugation reaction? Because what happens in type 2 reaction is, suppose this is your drug, now you have a conjugate, and this conjugate gets transferred into drug by an enzyme called transferase. When the conjugate gets transferred into drug by the enzyme transferase, then it forms drug conjugate complex, right? So this whole process is called conjugation reaction because of some conjugate or drug molecule, or some drug molecule, or any biological molecule that's present in the blood, right? Any molecule that is getting transferred into drug, that's called conjugate. So this conjugate is being transferred to the drug by the enzyme transfer, so which results in drug conjugate complex, and that's called conjugation reaction. So if this conjugate is glucuronide, what is glucuronide? Glucuronide basically means glucose-like molecule. So if this glucuronide conjugate gets transferred to the drug glucuronide complex is formed. If this glucuronide conjugate gets transferred in the drug, by the help of transferase enzyme, then drug and glucuronide complex is formed. So this is conjugation reaction involving glucuronide. And now what if in place of this conjugate glucuronide, what if there's acetyl group? What if there's acetyl group? If there's acetyl group, then the acetyl group is transferred into drug by the help of transferase enzyme, and that's drug acetyl complex. So this is new conjugate and drug complex that's being formed in this reaction. So this type of conjugation is acetyl conjugation reaction. Now there's another type of conjugation reaction that involves glutathione. So if the conjugate is glutathione, then the glutathione is transferred into drug by the help of transferase enzyme, and the new complex that is formed is drug glutathione complex. And this is the third type of conjugation reaction that we're going to study. Right, so out of all this, I just wanted to make clear is that phase 2 or type 2 reaction is called conjugation reaction because it involves the transferring of the conjugate into drug, and that forms drug and conjugate complex. And if the conjugate is glucuronide, that's called glucuronide conjugation. If that's conjugate is acetyl group, that's acetyl conjugation. And if the group is glutathione, that's called glutathione conjugation reaction. Right. Out of this, the three different conjugation reactions, the only important reactions from exam point of view are the glucuronide conjugation and the acetyl conjugation, which we're going to discuss now. So let's talk about glucuronide conjugation. So what is glucuronide conjugation? Let me draw that reaction again. This is drug, this is glucuronide, and in presence of this transferase enzyme, drug glucuronide complex is formed, and this is glucuronide conjugation. Now there are three important clinical vignettes that we need to discuss in glucuronide conjugation. That's the first one is chloramphenicol toxicity. Chloramphenicol toxicity. You know what? The chloramphenicol toxicity is due to overdose of the chloramphenicol, right? So the chloramphenicol in the babies, chloramphenicol is metabolized by enzyme called transferase, and it gets converted into metabolite, right? In babies, the enzymes involved in the chloramphenicol metabolism is not working in its full-fledged capacity. So the it cannot work properly. So the chloramphenicol may get accumulated in the blood of the babies, and it gives the baby called gray baby syndrome. So the enzyme involved in the chloramphenicol metabolism, that's transferase, is not fully functional in the babies, so babies present with gray baby syndrome. So this is one important clinical vignette you may be given. Now there's another clinical vignette that you might be given in the exam, and that's called bilirubin metabolism. Bilirubin is metabolized by the help of UGT1A1 enzyme. That's basically transferase enzyme. And when these enzymes are less in man, or these are not functional, you can get Gilbert syndrome, or you can get Crigler-Najjar syndrome, right? So this is the thing, or the clinical vignette they might give you in the exam. Now, next clinical vignette that might be given in the exam is use of phenobarbital phenobarbital in detecting or diagnosing type 1 and type 2 Crigler-Najjar. In type 1 Crigler-Najjar syndrome, what happens is there is total deficiency of that gluconosyltransferase enzyme. And in Crigler-Najjar type 2, there's only partial deficiency of the gluconosyltransferase enzyme. So what happens is, if you give phenobarbital is not only the inducer of the cytochrome P450 enzyme, but phenobarbital is also the inducer of the enzyme transferase UGT1A1. So the phenobarbital induces this enzyme, and more amount of the enzyme is produced, and this bilirubin, more amount of the bilirubin is getting changed into metabolite. So in Crigler-Najjar type 1, there is absolute deficiency of this enzyme. So this is adding phenobarbital in the patient's blood is not going to be effective in the patient. That's not going to be helpful. So when you add phenobarbital and the jaundice in the patient does not decrease, then you can be sure the type of Crigler-Najjar syndrome in the patient is type 1. But if you give phenobarbital and if it increases the enzyme, and if that increment in the enzyme decreases the bilirubin amount or that decreases the jaundice in the patient, then that means it has worked in the patient to some extent. It has helped the patient. So when the phenobarbital added in the blood, the patient helps the patient to get rid of the bilirubin to some extent, then you should be sure that is type 2 Crigler-Najjar syndrome in the patient. So this is all about glucuronide conjugation. Now let's talk about acetyl conjugation. So what's acetyl conjugation? As I told already, if there is acetyl group in the conjugate part of the reaction, conjugation reaction, that's called acetyl conjugation, right? So if this is your drug and if this is acetyl group in presence of this transferase enzyme, if you form drug and acetyl complex, that's basically acetyl conjugation reaction, right? So there's one important thing that you need to know. SLE, systemic lupus erythematosus. This drug can also be induced by some drugs. SLE can be induced or can be caused by some drugs like hydralazine or procainamide, or it can also be caused by isoniazid, right? So there is also something called drug-induced SLE, and the more common drugs that can cause SLE are hydralazines or procainamide or isoniazid, right? So there's a term called fast acetylators, fast and slow acetylators. So this drug-induced SLE is common in those patients who are called slow acetylators. So this is only the thing we need to know from exam point of view under the topic acetyl conjugation. So far, what we have discussed, let's review. So today, in this video, we talked about biotransformation, right? And we talked about definition of biotransformation, and we talked about different types of the biotransformation reactions, and we talked about the different types of type 1 reaction, that is oxidation, that's reduction, and that's hydrolysis reaction. We talked about some examples that involved in that type of reactions. Right now, we also talked about type 2 reaction, and the type 2 reaction are also called conjugation reactions. And we talked about 3 different types of conjugation reaction, and we talked about some clinical relevance of that reactions. So this is all for this video. We'll meet in the next lecture. Thank you.